Hello everyone, welcome to our own YouTube channel Shantanu Kolhe LifeScience. In the previous video, we have discussed internal Dudorov pathway. In this video, we will discuss anaerobic respiration and fermentation in detail. Okay, so this is also an average important topic from exam point of view because few questions have been asked from this topic in previous life science exams. Okay, so starting with the anaerobic respiration, the first important point which you should know is that both the terms anaerobic respiration and fermentation are different and they are not the same. Okay, anaerobic means this process will take place in the absence of oxygen. We all know this. Okay, it is different from fermentation, as I said earlier. So a question can be asked in upcoming exam whether anaerobic respiration and fermentation are same processes or they are different. Okay, so they are different. This difference is on the basis of terminal electron acceptor. Okay, so the final or terminal electron acceptor in anaerobic respiration is an inorganic molecule but accept oxygen, like nitrate and sulfate. Okay, in fermentation, this term was given by Cruikshank scientist and it is also known as zymosis by Pasteur. So this question was asked in previous life science exams. In Maharashtra set life science it was asked. Okay, fermentation is also known as zymosis, was reported or was given by which scientist? Okay, so the name of the scientist was Pasteur. Most of the students think that fermentation is anabolic process because they think that end product is going to be formed. Okay, but this is not the case. It is a catabolic process. In the first video, that is introduction to metabolism, we have discussed that anabolic process is those processes in which there is a formation of complex molecule from inorganic precursors, whereas catabolism is that process in which there is breakdown of complex molecule into simpler molecules. Okay, so it is a catabolic process. In fermentation, the final or terminal electron acceptor is organic molecule. This was also asked in CSIR net part C question because in unit 12, that is applied biology, we know that there is fermentation chapter. Okay, and from that it was asked whether the final electron acceptor in fermentation is organic molecule or inorganic molecule. Okay, so this was asked like 2 to 3 years before. Okay, in CSIR net exam. There is no electron transport chain process involved in fermentation, so this can be asked in upcoming exam. Okay, this statement is not asked yet in any life science exam, so it can be asked. And the substrate breakdown is extracellular. Okay, as it is catabolic process, the substrate will be broken down to produce final end product. Okay, in glycolysis, the substrate was glucose. Glucose was ultimately getting degraded into pyruvate, and glycolysis was taking place in cytosol, which means intracellular. Cytosol is in the cell, but in fermentation the substrate breakdown will be outside of the cell, that is extracellular. So a question can be asked from this statement also. So these are all important statements. Okay, the fermentation can be categorized into lactic acid fermentation, alcoholic fermentation. The names lactic acid fermentation and alcoholic fermentation comes from or are named on the basis of the end products which are formed. Lactate is formed, the name will be lactic acid fermentation. If ethanol is formed, the name will be alcoholic fermentation. Lactic acid fermentation will occur in liver, in muscles, lactic acid bacteria like lactobacillus, etc. In this step, pyruvate will get converted into lactate. When we know that in glycolysis, glucose will be converted into 2 molecules of pyruvate. That's why I have written 2 over here. Okay. Then when the fermentation of this will occur, these 2 molecules of pyruvate will get converted into 2 molecules of lactate by an enzyme lactate dehydrogenase. The cofactor is zinc ion and coenzyme is FMN. Okay. So what is happening over here? NADH plus H plus molecule, which is associated with lactate dehydrogenase, will be oxidized into NAD positive. We have been studying this in almost all of the videos, previous videos, that oxidation is loss of hydrogen atom and electron, reduction is acceptance of hydrogen atom and electron. So here NADH plus H plus is getting converted into NAD plus, which means it is losing hydrogen atom and electron, which means NADH molecule is getting oxidized into NAD plus, and this hydrogen atom will be accepted by this pyruvate, and we know that when the molecule will accept hydrogen atom and electron, we will say that it is reduced. So if this H plus, which have been released by the oxidation of NADH plus H plus, will get accepted by this pyruvate molecule as shown by this black arrow, we will get lactate, which means after the acceptance of hydrogen atom and electron by pyruvate, we will get lactate. So can we say that pyruvate is getting reduced to lactate? So pyruvate is getting reduced to lactate. This statement was asked in DBT JRF 2021 exam, that is conducted in this year. Okay, this question was asked. Okay, so now you can analyze what's going on over here. Okay, and it was not at all difficult. Okay, so you need to understand the process rather than just memorizing things, so that you can solve any question or any level question from any exam. Okay, now we will look at alcoholic fermentation. It will occur in yeast, that is Saccharomyces cerevisiae, etc. As discussed earlier, in glycolysis, glucose will get converted into pyruvate, and we will get 2 molecules of pyruvate. So that's why I have shown 2 written in black marker over here. Okay, so this is a structure of pyruvate, and we have also studied how to deduce the structure just by remembering 2 structures, that is glucose and fructose, while in glycolysis lecture. Okay, so if you are a new student, you can watch all the previous videos to get to grasp the concepts more clearly. Okay, so 2 molecules of pyruvate are getting converted into 2 molecules of acetaldehyde. Since 2 molecules of pyruvate are involved, 2 molecules of carbon dioxide will be released. Okay, so pyruvate will get converted into acetaldehyde by an enzyme pyruvate decarboxylase. The name itself suggests that decarboxylase means it will carry out decarboxylation reactions, means removal of carbon dioxide molecule. So 1 molecule of pyruvate can get converted into 1 molecule of acetaldehyde by the removal of 1 carbon dioxide molecule, but since 2 molecules of pyruvate are involved over here, 2 molecules of carbon dioxide will be released to form 2 molecules of acetaldehyde. I have also mentioned it, to decarboxylation of pyruvate to acetaldehyde will occur, and then acetaldehyde will be converted into ethanol. Again 2 molecules are involved, so 2 molecules of acetaldehyde will be converted into 2 molecules of ethanol. To convert 1 molecule of acetaldehyde to ethanol, 1 NADH plus H plus will be used, but since 2 molecules of acetaldehyde are involved, we will require 2 NADH plus H plus molecule to get converted into ethanol. Okay, so that acetaldehyde molecules can get converted into ethanol. Okay, this step will be catalyzed by an enzyme alcohol dehydrogenase. In this case, the cofactor is same, that is zinc ion, and coenzyme is TPP, that is thiamine pyrophosphate, which we have studied while discussing pyruvate dehydrogenase complex, that is and the first enzyme in pyruvate dehydrogenase complex, that is E1 or pyruvate dehydrogenase, also has TPP as a coenzyme. Okay, this is derived from thiamine or vitamin B1. Okay, so here there is also an oxidation of NADH plus H plus molecule. After the oxidation of this molecule, it will get converted into 2NAD plus. The hydrogen atom which is liberated from the oxidation of NADH plus H plus molecule will be accepted by acetaldehyde molecule, and when the acetaldehyde molecule will accept that hydrogen atom, it will get converted into ethanol. So this is the hydrogen atom shown by black arrow, which is accepted by acetaldehyde, and when the acetaldehyde will accept the hydrogen atom, it will get converted into ethanol. So the conversion of pyruvate to ethanol is two-step process, whereas conversion of pyruvate to lactate is one-step process. And there is reduction of acetaldehyde to ethanol, okay, because acetaldehyde is going to accept the hydrogen atom which have been released from the oxidation of NADH plus H plus atom, due to which after the acceptance of hydrogen atom by acetaldehyde molecule, it will get converted into ethanol, or it will get reduced into ethanol, okay. NAD plus which have been generated by this process, or NAD plus molecules are regenerated via this process, and it can again enter in the glycolysis in order to carry out the glycolysis under anaerobic condition, because we know that in the sixth step of glycolysis, 2 NAD plus molecules will get reduced to form 2 NADH plus H plus molecule, okay, and that is how we are getting 6 ATPs from glycolysis, okay. So to carry out the glycolysis, we need to regenerate the NAD plus molecules so that the glycolysis can continue, so that the NADH plus H plus molecule can form in glycolysis, okay. Now let us understand the energetics from where the 2 ATPs are coming from, okay. We know that in glycolysis, first step and third step, or we generally say that first, second, third steps are energy investment or priming stage or preparatory stage of glycolysis. In the first step, 1 ATP will be consumed, that is glucose to glucose 6 phosphate step, 1 ATP will be consumed. In the third step, another 1 ATP will be consumed, okay, that is fructose 6 phosphate to fructose 1,6 bisphosphate step, 1 ATP will be consumed. So in the energy investment stage, 2 ATPs are getting consumed. In the sixth step, NADH is getting formed. Which step is that? 1,3 bisphosphoglycerate step, okay. In the seventh step, 2 molecules of ATP will be generated, which is the reversible step instead of being the phosphorylation step. This we have also discussed while studying glycolysis. And in the 10th step, that is conversion of phosphoenol pyruvate to pyruvate, another 2 molecules of ATP are getting formed. So in the payoff phase or energy generation phase, we are getting total 4 ATPs and 2 molecules of NADH is getting produced. So 4 molecules are getting produced, but in the first and third step, 1 ATP is getting consumed. So 4 ATPs minus 2 ATPs give us 2 ATPs, and the 2 NADH plus H plus molecules which are produced in the glycolysis are going to be used over here. So we will not calculate it for energetics, okay, because we know that 1 molecule of NADH plus H plus will give us 3 ATPs, 2 NADH plus H plus molecule will give us 6 ATPs. But in the glycolysis, the 2 NADH plus H plus molecules which are produced will be used over here, okay. So the remaining ATPs will be only 2 ATPs. 4 ATPs minus 2 ATPs will give us 2 ATPs, and we will not consider ATPs from this, that is 6 ATPs, to calculate the energetics of anaerobic respiration, because these 2 ATPs, the 2 NADH molecules which are getting formed in glycolysis are getting used over here, okay, and similarly it will also get used over here. So the net ATP gain will be 2 ATPs, okay. So this can also be asked in the question, exam. Another thing, previously many times a question have been asked on the basis of these two terms, cofactor and coenzyme. The first thing you need to remember is that cofactor and coenzyme are both non-protein component. Okay. Second important thing is that cofactor is an inorganic molecule, coenzyme is an organic molecule. Okay. The role or the function of cofactor is to increase the rate of reaction or to make the enzyme function more efficiently. Okay. Whereas the main role of coenzyme is that it is also associated with enzyme, and its role is to serve as carrier to the enzyme, that is the transfer of chemical groups between enzymes and molecules. Okay. For example, coenzyme A can transfer acyl groups. Okay. And we also know that NADH plus H plus was coenzyme in many dehydrogenase enzymes. Okay. What it will do? It will transfer the hydrogen atom and electron between enzymes and molecules. For example, over here NADH plus H plus will be oxidized, due to which hydrogen atoms will be released, and that hydrogen ion will be accepted by the substrate to get converted into product. So the main role of coenzyme is to transfer the chemical groups between enzymes or molecules. Okay. So I hope this was very much helpful for you and all of the things are clear. Okay. So this is also important, okay, topic, you must go through it once. So I hope this was very much helpful for you. So if you found this video helpful, please don't forget to like, share and subscribe my YouTube channel. If you have any doubt, you can comment down in the comment section, and you can join my Telegram group. I will be providing the link in the description. It has more than 3000 premium quality quiz questions for your practice, and I also upload regular quizzes along with short notes, tricks about how to approach difficult part C questions, and I also upload regular important notifications about upcoming exams. Okay, so you can join that Telegram group. I will provide the link in the description. Okay, so see you in the next video. Thanks for watching.